Hi guys, welcome to today's video. In this video, we're talking about penicillin group of antibiotics. In the description below, you can find the link to the notes on this video as well as Anki flashcards on penicillin group of antibiotics. If you want to directly jump to each of the topic, you can click on the timestamp button in the description below. In this video, we'll talk about short history on penicillin, what is the basic structure of penicillin group of antibiotics, what are the different types of penicillins and their clinical use, formulations of penicillin, mechanism of action of penicillin, mechanism of resistance in penicillin, what are its adverse reactions, and a short description on methicillin-resistant Staphylococcus aureus. Before the antibiotic era, many scientists in the world were in search of a drug which can easily treat various kinds of infections. As we all know, it was Alexander Fleming who after multiple attempts had the breakthrough. He was working as a bacteriologist at St. Mary's Hospital in London, where he discovered the inhibitory effect of fungus Penicillium notatum on the Staphylococcus colonies. He published his work in the British Journal of Experimental Pathology in 1929. This sparked the interest in antibiotic therapy, and in 1939, scientists in Oxford started the research on the new found antimicrobial. This antimicrobial, eventually named penicillin, underwent cycle of refinement and purification. After many human trials, this antimicrobial was finally available for prescription in 1946 AD. For this discovery, Fleming, Florey, and Chen were awarded the Nobel Peace Prize in 1949, and this event laid the foundation of the discovery of many antimicrobials that are used in current era. Now let's talk about the basic structure of penicillin. Penicillin, like many other beta-lactam antibiotics, have a beta-lactam ring, which forms the backbone of this antibiotic. Cephalosporin, monobactams, carbapenems, and beta-lactamase inhibitors, they also have same beta-lactam ring in their structure. In addition to beta-lactam ring, penicillin also contains thiazolidine ring that is attached to the beta-lactam ring, and there is a secondary amine group attached to the beta-lactam ring. So basically, there is a beta-lactam ring, attached to it is a thiazolidine ring, and a secondary amine group is attached to the beta-lactam. The type of secondary amine group, that is R, determines the type of penicillin. What is the mechanism of action of penicillin? Penicillin, like every other beta-lactam antibiotics, they interfere with the transpeptidation reaction of bacterial cell wall synthesis. We should know that bacterial cell wall is different in gram-positive bacteria and gram-negative bacteria. In gram-positive bacteria, the cell wall is composed of peptidoglycan, and in gram-negative bacteria, in addition to this peptidoglycan layer, there is an extra outer membrane. And this extra outer membrane contains various channels, one of which is porin channel, which is very important, as this porin channel forms the transport channel for the penicillin antibiotic to enter into the periplasmic space. Now, this peptidoglycan is the site of action of penicillin. This peptidoglycan is a complex cross-linked polymer of polysaccharides and polypeptides. It is very important for maintaining cellular integrity, shape of bacteria, as well as it prevents the macromolecules from entering the bacterial cell. This peptidoglycan is composed of N-acetylglucosamine and N-acetylmuramic acid sugars. The characteristic strength of peptidoglycan comes from the peptide cross-linkages that is responsible for the net-like formation of peptidoglycan, which provides the strength to this peptidoglycan layer. These peptide cross-linkages occur between N-acetylmuramic acid residues. This cross-linkage is formed with the help of an enzyme called penicillin binding protein, and this process of formation of cross-linkage is called transpeptidation reaction. So what happens in transpeptidation reaction? A 5 amino acid peptide is attached to N-acetylmuramic acid, and this 5 amino acid peptide terminates in D-alanine amino acid. Now, the penicillin binding protein that we talked about is an enzyme. This removes the terminal alanine in the process of forming the cross-link between the terminal peptides of N-acetylmuramic acids. Penicillin, like various other beta-lactam antibiotics, are the structure analogues of D-ala, and this binds to the penicillin binding proteins, thus inhibiting the transpeptidation reaction. Now, since transpeptidation reaction doesn't occur, it halts the peptidoglycan formation, and this is why there is cell death of bacteria. Why penicillins do not affect the eukaryotic cells? As eukaryotic cells lack the peptidoglycan layer and the penicillin binding protein enzyme, they do not affect the eukaryotic cells. One important point we should know is that penicillin can act only on growing as well as multiplying bacteria, because only in growing and multiplying bacteria peptidoglycan formation occurs. Why penicillin activity is poor against gram-negative bacteria? As we mentioned before, the gram-negative bacteria, in addition to peptidoglycan layer in the cell wall, contain an extra outer membrane. Now, the penicillin, to reach its site of action, that is in the periplasmic space, where the penicillin binding protein is located, it has to pass through the outer membrane. The outer membrane contains various porin channels through which the transport occurs. Now, this porin channel can prevent the transport of penicillin to its site of action. That is why they do not act very well against gram-negative bacteria. Now let's talk about the categories of penicillin. Penicillin are mainly of three categories: penicillin G, antistaphylococcal penicillins, and broad-spectrum penicillins. Let's talk about penicillin G. Penicillin G, also known as benzyl penicillin, was the first molecule extracted from the Penicillium notatum. Penicillin G are highly active against gram-positive cocci, gram-positive rods like Listeria. They are also active against gram-negative cocci such as Neisseria. They are active against most of the anaerobes like Clostridium. And they are also active against spirochetes like Treponema pallidum. However, they are not active against beta-lactamase producing Staphylococcus. They are not active against resistant group of Pneumococcus. Also, they do not act against some enterococci and methicillin-resistant Staphylococcus aureus. They are not active against gram-negative rods, and they act very poorly against anaerobes like bacteroids. Penicillin G is only bacteriostatic against enterococcus, and there is rising resistance against penicillin G. So, if we talk about formulation of penicillin G, benzyl penicillin is the intravenous formulation. It is also available in intramuscular forms as procaine and benzathine penicillin G. We know that benzathine penicillin is used as prophylaxis against Streptococcus pharyngitis for prevention of rheumatic heart disease. It is also used in treatment of syphilis. Is there any oral preparation for penicillin G? Yes. Penicillin V is the oral preparation of penicillin G. It is only used in minor infections because it's very poorly bioavailable. It requires very frequent dosing and it has very narrow spectrum of activity. Now we talk about antistaphylococcal penicillins. So after the introduction of penicillin G, it worked very well in the beginning, but gradually there emerged a group of bacteria which had the capability of producing lactamase enzyme. This lactamase enzyme acts on beta-lactam ring and break it down, producing penicilloic acid, which is very ineffective antibiotic. This led to the development of semi-synthetic beta-lactamase resistant penicillins. These are the antistaphylococcal penicillins. They are active against staphylococci that produce beta-lactamase and streptococcus. However, they are not active against enterococci, anaerobic bacteria, gram-negative rods as well as cocci, and methicillin-resistant Staphylococcus aureus. This means that they are active against the beta-lactamase producing organisms, but they are very poorly active against enterococcus, anaerobic bacteria, gram-negative rods as well as cocci, and MRSA. These drugs include methicillin, nafcillin, cloxacillin, dicloxacillin. Methicillin was the first antibiotic in this group. However, it is not used these days because of significant adverse effects. Now, since these antistaphylococcal penicillins were only active against beta-lactamase producing Staphylococcus and had very poor spectrum of activity over the gram-negative bacteria, this led to the discovery of new kind of penicillins which were active against gram-negative organisms as well. The broad-spectrum penicillins, they have greater activity against gram-negative bacteria as they have better ability to penetrate the gram-negative outer membrane. But like penicillin G, these drugs are easily inactivated by many beta-lactamases. This category includes three subcategories. They are aminopenicillins, carboxypenicillins, and uridopenicillins. The aminopenicillins include amoxicillin and ampicillin. Both these drugs have very similar spectrum of activity, but amoxicillin is better absorbed orally. Both these drugs are most effective of all beta-lactams against pneumococci. Ampicillin, but not amoxicillin, can be used against Shigella, and they are active against majority of gram-negative organisms like E. coli, Salmonella, Shigella, Protease, and non-beta-lactamase producing Haemophilus influenza. But many resistant cases have emerged, and nowadays these drugs are rarely used in case. The major disadvantage of this subcategory of drugs are they are not active against Pseudomonas, Klebsiella, Enterobacter, Serratia, Citrobacter, indole-positive protease, and other hospital-acquired gram-negative organisms. Now, the second subcategory in broad-spectrum antibiotic is carboxypenicillin. Carboxypenicillins include carbonicillin and ticarcillin. Carbonicillin are the first anti-pseudomonal antibiotic, but now it is not used. These have activity against Pseudomonas, Protease, and Enterobacter. Ticarcillin is also carboxypenicillin and has similar spectrum of activity like carbonicillin, but they are 2 to 4 times more active on a weight basis against Pseudomonas aeruginosa. Ticarcillin is often used in combination with beta-lactamase inhibitor like ticarcillin clavulinate. What is the problem associated with ticarcillin? Ticarcillin is a disodium salt, so it cannot be used in case of volume overload. Also, it inhibits platelet function, thus prolongs bleeding time and causes bleeding diathesis. What are uridopenicillins? Piperacillin, azlocillin, meslocillin are uridopenicillins. These are often considered as 4th generation penicillins. These drugs are better than previous drugs against Pseudomonas. They have better activity against Klebsiella, but cephalosporins are always preferred. Piperacillin, however, is found to be ineffective as single agent against the growing resistant groups of organisms. Thus, it is used in combination with beta-lactamase inhibitors in the form of piperacillin tazobactam. This increases the spectrum of activity as it covers both beta-lactamase producing Staphylococcus and beta-lactamase producing gram-negative organisms. Now let's talk about the mechanism of resistance that the bacteria develops against penicillin. Resistance to penicillin develops by one of the following 4 mechanisms, either by inactivation of antibiotic by beta-lactamase enzyme, or by modification of target penicillin binding proteins, or because of impaired penetration of drug to target penicillin binding proteins, and by antibiotic efflux mechanism. Inactivation of antibiotic by beta-lactamase enzyme is the most common mechanism of resistance. This enzyme breaks down the backbone of an antibiotic, that is beta-lactam ring, producing penicilloic acid, which lacks the antibacterial activity. Various types of beta-lactamase enzymes are present and they have specific action. For example, some beta-lactamase they hydrolyze only penicillin, but they do not have any effect on cephalosporin. AMPC beta-lactamase produced by Pseudomonas and Enterobacter and extended spectrum beta-lactamases hydrolyze both penicillin as well as cephalosporin, whereas carbapenems are resistant to hydrolysis by both penicillinases and cephalosporinases but are hydrolyzed by metallobeta-lactamases and carbapenemases. The second way of development of resistance is by modification of penicillin binding proteins. Third mechanism is by impaired penetration of drug to target penicillin binding proteins. This mechanism occurs only in gram-negative species because they have the impermeable outer membrane of cell wall, which is absent in case of gram-positive organisms. Now the fourth and final mechanism is by antibiotic efflux mechanism. That is, this also occurs in gram-negative organisms and they contain various efflux channels that pumps out the antibiotics from the periplasmic space to the outside of the cell. Now let's talk about units and formulations of penicillin. Crystalline penicillin G contains 1600 units in 1 mg. And that is 1 unit contains 0.6 microgram and 1 million units contain 0.6 gram. Most penicillins are formulated as sodium or potassium salts of their free acids. Thus, we should be careful in case of hyperkalemic or hypernatremic. What is the pharmacokinetics of penicillin group of antibiotics? Almost all the penicillins can be administered orally. However, there is problem with nafcillin as their absorption is very erratic. Penicillins usually have shorter half-life except that of intramuscular preparations. Also, piperacillin has a dose-dependent pharmacokinetics and a longer half-life is obtained when higher doses are administered. Penicillin group of drugs are widely distributed throughout the body but not in the CSF. However, in case of CNS infection where meninges are inflamed, they can easily enter the CNS when given at higher doses. How penicillin is excreted from the body? Penicillins are excreted via the renal route. Tubular secretion accounts for about 90% of the excretion and remainder is by glomerular filtration. Almost all of the drugs require dose modification in case of renal insufficiency. However, nafcillin, oxacillin, cloxacillin and dicloxacillin have major non-renal routes of clearance and they do not need dose adjustment. Now let's talk about the side effects of penicillin. Penicillin is generally well-tolerated drug and most of its side effects is due to the hypersensitivity reaction. Allergic reactions like anaphylactic shock, serum sickness and skin rashes can occur. Also, there are few rare side effects like if you give penicillin in very high doses in case of renal failure, they can have seizures. Nafcillin can cause neutropenia, oxacillin can cause hepatitis, methicillin can cause interstitial nephritis, and one very important side effect is by ampicillin that it causes pseudomembranous colitis. Now let's talk in brief about MRSA. We'll talk about this treatment in our upcoming videos. Here we'll just talk in short about MRSA. MRSA is methicillin-resistant Staphylococcus aureus. Methicillin was a semi-synthetic antibiotic that was introduced in 1959 against beta-lactamase producing Staphylococcus. But in the following year, that is in 1960, there was a major outbreak of MRSA in Europe. MRSA has now emerged as a major nosocomial and community-acquired pathogens. Now, how can we label Staphylococcus as a MRSA group of Staphylococcus? Methicillin resistance requires the presence of MEC gene, and with the help of polymerase chain reaction, we can identify the MEC gene. As per the Clinical Laboratory Standards Institute, MRSA is defined as oxacillin minimal inhibitory concentration of 4 or more than 4 microgram per ml. Oxacillin is used for defining MRSA, not methicillin, as methicillin is no longer used these days. Now let's talk about MEC gene. The structural component of MEC gene is MECA. This encodes for the penicillin binding protein 2A, which is different from the normal penicillin binding protein where penicillins attach and prevent the transpeptidation reaction. This MECA gene is responsive for resistance to the entire beta-lactam group of antibiotics except for ceftaroline and ceftobiprole. Apart from MECA, other homologous genes like MECB, MECC, MECD have also been identified. Now, how this MEC gene gets transferred from one staphylococcus to other staphylococcus? MEC gene is carried by a mobile genetic element that is present within the staphylococcus, and it is called as staphylococcal chromosomal cassette MEC. There are about 13 SSC MEC identified till date. Type 1, 2 and 3 SSC MEC are associated with nosocomial MRSA and are multi-drug resistant, whereas type 4 and type 5 SSC MEC are associated with community-acquired MRSA, and they are less notorious compared to hospital-acquired MRSA, and they produce a PVL toxin. This SSC MEC is transferred horizontally from one Staphylococcus aureus to another Staphylococcus aureus, and in this way they lead to the development of outbreak. With this, we have come to the end of this video. For more videos, kindly subscribe and hit the like button so that you can get the notification whenever I upload the next video. In the next video, we'll talk about cephalosporin group of antibiotics. Thank you.